We've been talking about how microbes are beneficial to us in a number of different ways, and wrapped up with food poisoning and how there were two types of food poisoning, one being intoxication from the exotoxins that the bacteria secrete, or other microbes that they secrete, or the actual foodborne illness where that food contains um, microbes that are still active and able to infect us. And then we wrapped it up with talking about the different methods for preventing foodborne illness, which were things that we had already discussed in the previous chapter, the types of microbicides and microbiostatic techniques to keep germs at bay. All right. So then what I wanted to shift into now is how we use microbes from an industrial perspective to make things for us, whether it's natural metabolites and byproducts of microbes, such as antibiotics that they use to kill off other microbes, or certain vitamins that they make that we don't, or that we need more of, or if it's things like acids or solvents that we use for whether it's acetic acid for vinegar, or alcohol in the fermentation process to either consume or to use as a germicide, or enzymes. Lots of different things are made by microbes in an industrial setting that have been designed to come with some kind of product for us to utilize. Oftentimes, this is going to involve fermentation of some sort. And so here are some enzymes that are commonly made in an industry setting. Amylase. Amylase was actually the first enzyme ever discovered. And anybody know where amylase is? What enzyme that is? What it's used for?
I'm just gonna guess, is it to cleave off? Oh yeah, somebody wrote it. I think they're right, saliva.
Yeah, so salivary amylase is kind of its full name. And um, it's an enzyme in your saliva that helps you break down your food. So as you masticate and chew your food, the enzyme then, and you're moving it around into that bolus, the enzymes start breaking that stuff down. So before it even gets to your stomach, it's already starting that process of breaking down that food. And it's got all different types of applications outside of our own digestion. Okay. And it can be used in a lot of different food preparations, and even in the textile industry. Cellulase. Cellulase is something that humans lack. Cellulase is an enzyme that breaks down cellulose, which is kind of like a complex sugar in a lot of plants, which is why this is why we don't eat grass. This is why your dogs, if they do try to eat grass, it will either come out undigested or it will come out of their mouth. They'll throw it up because you can't break it down. We don't have the enzyme cellulase. Cows and horses. Yeah. Yes. They do have cellulase, and so they can break down these things. It was used, interestingly, for stonewashing. I don't know if anybody knows what stonewashing is. I mean, I guess 80s are kind of making a comeback, but it's a type of treatment to genes that was very popular in the 80s. Also used as a digestive aid, again, because we consume things that have cellulose in them. Hyaluronidase, used for different wound cleansings, and keratinase, That's for things like hair removal from hide. So in the tanning process, keratinase breaks down keratin, and keratin is the stuff that's in our fingernails and our hair. Pectinase is for breaking down things that have pectin, which is mostly fruit. So if you want to make jam, for instance, you typically you add pectin to it, and that gives it its kind of jelly-like consistency. Proteases, we've talked about proteases. They break down proteins. And so one example of a protease would be like a meat tenderizer, right? So you put this white powder on meat and it starts breaking down the proteins in that meat to soften it. And oftentimes these proteases come from bromelain, which is a protease found in pineapple, which is also why a lot of people, when they cook ham or something, they'll put pineapple on it. And the pineapple serves, I guess, to give it flavor too, but also the pineapple helps tenderize the meat. Okay, so these are just a smattering, just a few of the enzymes that are produced industrially using bacteria, mostly, and their sources, but not all. Some of these are molds that are bent to the will of industry. How do we go about doing this? Typically, we genetically engineer bacteria or fungus to produce large amounts of a certain metabolite. And it can be a primary metabolite or a secondary metabolite. Well, what's the difference? Primary metabolites are produced during that major metabolic process, okay? It's essential to the microbe's function. So remember the paper we discussed last week when they were changing that pasture P from a chemotroph or from a heterotroph to a chemotroph, and they had to alter that pathway. That would be a primary pathway, and therefore the byproducts of that, one being formaldehyde initially, would be a primary metabolite. Secondary metabolites are byproducts that aren't critical to the microbe's function, okay? Still may be beneficial to the microbe, but not essential. So certain vitamins, certain antibiotics, certain steroids, okay, during the stationary phase, when the population isn't actively expanding, they're just maintaining. And so industry will tweak either primary or secondary metabolites depending on what end product they want. And so this is looking at a diagram of the primary and secondary metabolic processes and where that fits into the growth phases of the bacteria. So we remember lag phase, and then exponential phase when it's rapidly dividing, or log phase, and then it plateaus off to stationary phase, and then eventually that population starts to die off, right? And so we can expect primary metabolites to be produced during this exponential phase. They're essential to the expanding of the population, essential to the cellular division of these bacterial cells, for instance, or these yeast cells. And so if you wanted to harvest a primary metabolite, you would want to be doing that when the culture is in the exponential phase. That's when it's going to be kicking off a lot of these primary products. Now, let's say you wanted to harvest something that was later on in the secondary phase. You would wait till that population reaches a stationary phase. And you can control that by the nutrients that you put into that culture, right? If you want to get to a stationary phase, then you would make sure that at some point they start to exhaust those nutrients and then stop their division. And then they start making these byproducts that you can then purify out of the culture and then use. Okay, any questions on that? There are two different kind of ways in which industry can get these products. One is a batch fermentation. So you add your substrate, whether it's, let's say it's just sugar, for instance. You add it to the fermenter or the bioreactor, and you go through a limited time process until you get as much product as you can or want, and then you're done. And then you remove everything out of that bioreactor or that fermenter, and you clean it out and you start over again. That's a batch fermentation. A continuous feed system is that your reactor, you don't really, I mean, occasionally you might take everything out and clean it out just to, and do decontamination every so often. But essentially what you do is you just add nutrients continuously, and you continuously then siphon off the products that are made from that. So for instance, when I make my kombucha, I use a continuous feed system. So I have a big jar, and at the bottom of the jar, I have a little spout, okay? And then inside the jar, I have the SCOBY, that biofilm stuff. And I will take some tea and put a bunch of sugar in that tea, so make good old Southern sweet tea, pour that into the jar. And then once it's fermented, I draw it off, enough to bottle up, but I keep some in there, and then I start over again. And I pour new sweet tea into there, and it just does it continuously. And then occasionally I'll take the SCOBY out and I'll wash it all off on the inside with soap and water. So that is a continuous feed system. And that's the difference. Batch fermentation is just like a one and done. Continuous is using the microbes in that reactor continuously, at least for a certain period of time. Some more examples of products that we make using microbes in industry. Well, antibiotics, right? Obviously, hormones. So, for instance, when we have certain deficiencies, there are all kinds of hormone therapies that are provided to patients, right? And it used to be that those hormones were purified from oftentimes pigs. But now we can just take the gene that makes that hormone and insert it into a bacterial plasmid, and then have the plasmids transcribe that RNA off of the plasmid, and then translate into protein, and then we extract it, purify it, and we have a lot more than what we could get from pigs. Certain vitamins, because remember, bacteria, when we talk about the microbiome, we've already kind of hinted at this with our paper that we did for the first journal club, but bacteria break down certain nutrients into vitamins. Okay, and that's beneficial to us in the absorption of the microbes in us, but then also to make vitamins that they sell on the shelf. That comes oftentimes from microbial produced sources. And then definitely vaccines. Biopesticides. So what's a biopesticide? That is taking either a pesticide that a microbe uses, or more commonly, using a microbe to produce a certain pesticide, rather than chemically making the pesticides in like an organic chemistry lab. For instance, with the BT cotton, right? They put this microbe gene. They use a microbe to put a gene into the cotton that then produces a substance that is toxic to pests of the cotton. Okay. Certain amino acids, right? So if you are supplementing with amino acids, like branched chain amino acids or essential amino acids. It can be purified from things like whey, which is dairy, but it can also be made with, again, microbes. Natural flavors, solvents, organic acids, these kinds of things are typically made using microbes in industry. Okay, and so this is a table. You don't have to memorize this stuff, but it gives you an idea of where our things come from, what source. So cephalosporin, that's not too much of a stretch that it comes from Cephalosporium yeast. And then penicillium also, or I guess these molds, right? Vitamin B12, it's important because we don't make it, we have to consume it, and we can get that from Pseudomonas. Steroids, citric acid, acetic acid, so on. Okay, so just a few examples of this. Ethanol, acetone, so these are going to be our solvents. Gives you the different microbial source, what it can be used for. So just review this if you want, and it's not going to be stuff you'll be tested on. No quiz this Friday, because this is, again, kind of a more casual week of lectures. So I'm not going to give you guys a quiz for Friday on this stuff. Okay, any questions before we get into the last little section or comments? No? All right. Last little bit I want to talk about is using microbes and biotechnology. Okay, so several years, well, when I say that, I'm talking, I guess, more like 15 years ago, there was a lot of promising use of adenoviruses or lentiviruses for gene therapy. Okay, so taking a virus and programming it to have a certain gene that it carries that will then insert itself into the genome of the host to correct a mutation. Essentially, that was the idea behind it. And there was a lot of hype behind it, and it's actually used in research a lot to take in cell culture to program cells. You can use lentiviral vectors and manipulate them, but they were actually experimenting on this lentiviral delivery system on humans. And there was a really unfortunate event that happened at, I think, UPenn, where a young male in his 20s, I believe, died as a result of lentiviral treatment. And so that really put the brakes on this as a source of genome editing in humans. And so they still, like I said, it's still used in research on cells and stuff, but not really so much as a gene therapy in genetic diseases because of that situation. But instead, now we have a new technology that is also using a microbe as a delivery system, or not using a microbe, but using something that a microbe has, and that's CRISPR. And you guys will remember that last month when they were announcing the Nobel Prizes for chemistry, Emmanuel and Jennifer, and I don't know them, so I guess I should use their full names, but they were both awarded jointly the prize for their adaptation of CRISPR into an application. Okay. And it was big. It was great because it was first of all, two female scientists had never been both awarded the Nobel Prize in chemistry jointly. And so that was awesome. And then it was also pretty cool because CRISPR hasn't been around that long. I mean, we're talking about something that was applied like this about 8 years ago, and usually it's decades before scientists get recognition for their contributions. But so now CRISPR is a very promising genome editing tool, also used in research on cells and culture or on model organisms, and is slowly being adapted to humans as well. So what I wanted to do is I wanted to play a short little animated video on CRISPR. So for those of you who aren't familiar with this process, we'll have a little bit more knowledge. Whoop. I guess I didn't embed that as a link, so hold on. Any comments or things about CRISPR you want to mention? It's exciting stuff. Oh, easy. Let's do this. All right, now let's see if I can play a video.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene, or introduce a stop codon at a specific place. But it's not all about gene editing. Several labs have been working on ways to use CRISPR to promote gene transcription. They do this by deactivating Cas9 completely, so it can no longer cut DNA. Instead, transcriptional activators are added to the Cas9 by either fusing them directly or via a string of peptides. Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery, bringing RNA polymerase and other factors to the target, and increasing transcription of that gene. The same principle applies to gene silencing. A CRAB domain fused to the Cas9 inactivates transcription by recruiting more factors that physically block the gene. A more outside-the-box idea for using CRISPR is to attach fluorescent proteins to the complex, so you can see where particular DNA sequences are found in the cell. This could be useful for things like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus. CRISPR has already changed the face of research, but these new ideas show that what's been achieved so far could just be the tip of the iceberg when it comes to CRISPR's potential. Whatever comes next, it seems the CRISPR revolution is far from over.
All right. So, really very cool stuff, in my opinion, and it's not going anywhere soon. We're just beginning to be able to modify this CRISPR-Cas system to benefit humans. And you see the different applications, right? It's not just genome editing, it's also genome labeling with fluorescence. And so one thing I want to mention, they talk about fusing different proteins onto that CRISPR Cas-base substrate. How do you think they do that? How are they changing that CRISPR protein? What are they using to do that? Anybody know?
Well, it's clear right in the picture that you're showing a pair of scissors. Come on now.
Not quite. They get a little tiny human. No. So they use bacteria. They use the plasmids that I was talking about, and they insert the gene that would make this caspase enzyme. Only they add on to that new sequence of DNA that now allows that to be modified. Okay, or they tack on new sequence of DNA that gives that caspase a sticky site for some enzyme to be recruited and bind to it. Okay, so again, we are using industrial methods. We're using recombinant DNA techniques with bacteria to take this genome editing tool that came from bacteria and use it to benefit humanity. Okay. So again, highly, highly beneficial things coming from microbes. And any kind of alteration in a genome sequence is typically done using bacterial plasmids. Now, what do we know about CRISPR? Well, we know that the reason we're not using it right now on humans is because there are all these off-target regions that the CRISPR can bind to, okay? Because that RNA guide is such a small segment, it can have complementarity to a bunch of different sites in the human genome. So you may want to be targeting one specific mutation, but as a result, it binds to that and a bunch of other downstream or upstream genes and could potentially silence them or correct them, but it's not really correcting it, it's altering it. And so one of the biggest obstacles to overcome is to be able to take this CRISPR and make it more specific. And this paper came out a couple years ago, and they have done just that. They have essentially modified this caspase-9 protein so that it can work with less off-target binding. Okay, and so it's still not foolproof, but it's being used in different things like detection systems. So remember in that video, it mentioned using the fluorescent tag and then just have it bind to a certain sequence. Don't do any cutting necessarily or editing. It just binds to that region and it has a little fluorescent molecule attached to it, and then it lights up. And so earlier this year in April, in Nature Biotechnology, they used a CRISPR-Cas12, not Cas9, because it won't cleave anything, but it will have that molecule, that fluorescent molecule that they can then use to detect the presence of SARS. And it's supposed to be much less false negative, so a 95% positivity and 100% negative predictive agreement. So that means there are no false positives, and there are almost completely 100%, 95% true positives. Okay, which is huge because the system, if you guys don't know, the COVID test that I got to go do for my 2-year-old in about a half an hour is not nearly that robust. And it also depends on at what point the person is infected. And when you catch that, you know, if you test them too early or too late, you'll get varying degrees of results. But so this clearly hasn't been adopted widespread in this country. But it's using CRISPR for something that's very important for surveying another microbe, that being a virus. With that said, in June, this paper came out saying that CRISPR editing in human embryos wreaks chromosomal mayhem, which is a nice, colorful title. But there were 3 different studies showing large DNA excisions from these embryos. And so again, these off-target sites being manipulated without the intent of that happening. And so a couple months later, they came out with this decision that CRISPR babies are still too risky. It says influential panel. So we have still a long way to go before we can start using this CRISPR on humans, whether it's on embryos to correct familial mutations. You know, if you have a family history of a certain mutation that can predispose you to whatever disease or illness, and it's not like you can necessarily screen embryos for that mutation. Let's say it's going to be in all of them. Potentially this could be used someday to fix that. Potentially this could be used not on embryos, but on fully developed humans, whether juvenile or adult, and correcting things that otherwise would be a lifelong discomfort or, you know, a premature death. So that's all I have for biotechnology. That's all I have for this chapter. No quiz this Friday. What I want you to do is focus on your proposal, your draft, okay? That's due this Friday. We'll lecture Friday on the microbiome a little bit, but no quiz. And what I want to do is use the rest of this hour to talk about proposals. So if you feel comfortable where your proposal is and you want to just start working on that, you're welcome to sign off. And those of you who want to stick around and chat, I'm happy to do that. There's a Labster that I've published, so remember to do that as well. If you are doing well on your proposal, you want to rock that out, by all means, it's up to you to use this time as you want. Any questions on that chapter or comments or anything else related to this class before we move on to the next thing?
Hey, Doc, do you know what the acronym, or I guess it'd be the abbreviation, CRISPR stands for?
Yeah, let's see. So it's like, no, I don't. What is it? You have it looked up?
No, man, I was putting all my money on you, buddy. What happened?
No, I mean, I don't know. It's like Caspase. No, what is it? Anybody else know it? It's something like um,
Does the C stand for clustered?
yes, clustered, clustered, uh, hold on. Clustered, regularly interspaced, short palindromic repeats. I think that's it. Short palindromic repeats. So does that make sense? CRISPR? That's what I think that's what it stands for. Look it up and double check. Any other questions? Clustered, regularly interspersed, short palindromic repeats. I think that's it. It's close anyway, if it's not. Yeah, isolation problem. Okay. So there's a problem with the Labster bacterial isolation simulation, being able to pick up a test tube from the box. Did anybody else have that issue?
No, but I had an issue on another Labster and I reached out to like the help site or whatever, and they just told me to like go back under the mission tab and you can like reload a checkpoint and it will take you, you might have to redo a few things, but it will take you back. And then if you're stuck, like that allowed me to like get through it. So I would suggest maybe trying that.
Okay. Good. Also, growth on your plates. That's been a few of, has anybody had any growth? Let's start off that way. Have any of you been able to culture from yogurt? No, I'm assuming nobody has. Nobody has any cultures on their plate? Okay. So the most important thing is that we just have growth on there. And the yogurt was used because it's non-pathogenic and it does have bacteria. And so the idea is that it would be a great way to get bacteria on your plate so that you can then test different antiseptics and disinfectants. So if you are not able to, if no one has any growth from this yogurt, then maybe there's something wrong with the yogurt. I don't know. What I recommend, oh, here's a plate. Oh, you got some stuff on there, Ryan. Okay, great. What I recommend is if you can't, I'm going to extend the due date. Okay, so don't worry about turning it in by Friday. What we can do is we can just let it go a little bit further until you get some growth. And if you're not seeing any growth with the yogurt, then what I recommend is just doing something else to get, all you want to do is get bacteria on there. So if you have to cough on it, or if you want to swab your sink and, you know, just run it around where the garbage disposal is, or like down into the drain area, that's going to have lots of bacteria. If you want to rub a Q-tip, even doesn't have to be a sterile Q-tip. We just want to get bacteria on there. Okay. So inoculate the plate with something and then get it to grow. Once you get some growth on there, then soak your little discs in alcohol and detergent, or you can get experimental. You know, if you say, hey, I want to test mouthwash, or I don't know, maybe you want to test some kind of tincture or essential oil, you know, that you think might be antimicrobial, that's fine. I mean, I want you to do at least something that has been proven that it does have strong microbicidal qualities so that you have a control. But if you want to try something else that is less scientifically proven, by all means, go for it. Okay. I'm not looking for you guys to, you know, here's the thing. I'm going to get philosophical for just a minute here. When you guys do these labs, oftentimes they're labs that are picked because they work. They're reproducible, they're successful, and that demonstrates things that we talk about in lecture, right? But that's not how science really works. And when you do these types of labs, it encourages this idea that that's what science is. You show up, you do what it says in the protocol, and you get a result, and then you get a good grade, right? Science is curiosity. Science is creativity. Science is determination. Science is outside the box, okay? And if you guys do that stuff and you still don't get an answer, it's better than doing it all the right way and not getting an answer either, right? So I encourage you guys in this class, the way it's set up, to get creative, okay? And you're not going to be penalized from that. You're going to get rewarded for that, if anything. So with these kits, if things don't work, come up with a solution. And if that doesn't work, no biggie. But you're using the scientific process that way. You're troubleshooting. Okay. So get off the soapbox right there. But that's my encouragement to you guys. That's my task that I give to you guys. Because you're doing this alone, you're doing it at home, and you're doing it with very little guidance. And I understand all of that. Okay. So we'll extend the due date. Not a big deal. Just get some stuff to grow. Okay. And then try to kill the stuff that grows. And then it's done. Okay. Okay. Now moving on. Anybody want to talk about their proposal? Anybody need some help or guidance with their proposal while we still have a little bit of time?
Actually, I wanted to just ask a question, a clarification question. So if we don't have growth on our plate, should we just reuse that plate, or would you
Yeah.
be able? Yeah.
Just reuse it.
Okay.
If there's nothing on it, then it should be fine, right? And do it sooner
Oh,
than later,
right.
because we don't want the plates to dry out, right? And we know that they
Right.
dry out. Yes. So I know that some of you guys wanted to chat, so now is your time to talk about your proposal and what you want to do. Okay. Luce, I think you wanted to talk, right?
Yeah.
So what is your, remind me what your project is so far?
Well, I just want to do something with H. pylori.